Greetings. I know many of you, maybe most of you. I'm Marshall Runge, and I'm just giving a brief introduction today. This is the second of our in-person town halls after our COVID experience. And as with the last one, the goal is to make these very interactive. And we'd love to hear from you. We'll spend about half of our time talking and giving some very interesting presentations, and then look forward to hearing your questions, thoughts, concerns. So thank you very much for being here. We will have Keith Dickey, who's our expert discussion leader, Q&A guy throughout the pandemic. Will be fielding questions and helping. And then we have some three speakers, but first I'm going to turn it over to Carl Jepson, who's Associate Dean for Research. Carl is also a professor in the Department of Orthopedics, and Carl's going to give some overview comments, and then we'll introduce our guest speakers today.
Thank you, Dr. Runge. So welcome, and thanks everyone for taking the time to come out today. I am standing in for Steve Kunkel, who could not be here today. So as you can see from the agenda, I'm just going to give a very brief update on the Great Minds, Greater Discovery Strategic Plan. Introduce three faculty who agreed to give TED-like talks about their research and to keep, going back to the idea that research is fun, and then we'll move on to the Q&A portion. So I'm gonna give you a series of numbers. It's not the Fibonacci sequence, nor is it next week's lottery numbers. 724, 6.5, 65, 64, 51, and 1. 724 is the number of millions of dollars we have spent on externally funded research for the past year. It's a 73% increase in research expenditures over the past 10 years. So pat yourself on the back. This is amazing. We've recovered from the pandemic pretty well. 6.5 refers to $6.5 million of support provided to 65 faculty and 64 trainees. This is part of the Pandemic Recovery Program. And the PRR was a 3-year program. It was a partnership between the Office of Research and the departments. And this is one of the things that we're probably most proud of, is the fact that when our faculty and trainees are down, we are behind them. So so we all suffered in many ways from the pandemic. Some faculty just had a harder time getting back on their feet. So this program was remarkable. Again, it's a partnership between the Dean's Office and the departments, and it's a cornerstone of the strategic plan, and that's people. Okay, and it really worked. So the faculty and trainees are back on their feet. We're keeping track of these, and we've already had 20 grants funded as of today. 51 is the number of med school faculty supported through the Scouts program. Now, we've been advertising that quite a bit because we're trying to promote another cornerstone of the strategic plan, which is bold science. We're asking our faculty to think way beyond, okay? And the Scouts program supports these crazy serendipitous ideas that have just been sitting there, waiting for someone to give them a little bit of support, a little bit of a boost to pursue answers to these questions. And 1 is the number of individuals awarded the National Medal of Science. This is Dr. Huda Akil. I don't know if she's in the audience, but she's a neuroscientist who has explored the brain secrets for over 50 years. So she was awarded the National Medal of Science from President Biden this past year. Okay, so this is just a small flavor of the Great Minds, Greater Discovery Strategic Plan, and I want to give a little bit more about what's on the horizon. Okay. So at the NCRC site, this is still work in progress. They're still in the planning stage of building out the Innovation Hub. This may include a new hotel and conference center and new research buildings. Infrastructure, our favorite topic. Okay, so all of our space is nearly programmed out. You still see a few empty labs, but they are programmed out. It's taking us a while to fill those in, but we are working on a new plan. Okay, the new plan should provide an acceleration of our ability to allocate space, more certainty in planning for our chairs and CDAs to recruit and support our existing faculty, but also to establish standards that we hold all of our departments accountable to. Gen AI, a new emerging area of interest to all of us. So task forces are being assembled to develop principles for the use of Gen AI. We have Brian Athey in the audience, and if you have any questions, you can speak to him. This is in trying to define some principles and guidelines about the use of generative AI in our research, education, and academic missions. And the last update is you've been getting a lot of emails from Brad Martin and his team in FFMI, which is Fast Forward Medical Innovation, and trying to encourage more departments to capitalize on the resources available to commercialize and work with industry partners. Okay, and I just want to speak very, very quickly to the statewide expansion. There's a lot of details here, so I'm just going to read this straight out. So, and this is statewide expansion as it pertains to research. So as Michigan Medicine develops a growing number of affiliations with healthcare systems around the state, this introduces new opportunities for partnerships and research. This provides opportunities for patients around the state to participate more easily in clinical trials. It's a key benefit of these partnerships and integral to the overall vision of the U of M Health statewide network of care. The details of how collaborations and research would work vary based on a variety of factors, including the nature of the relationship between U of M and the affiliate, the study's characteristics, and the specifics of the physician or faculty PI's appointment at U of M or the affiliate. U of M faculty should be aware that they may not extend research to any affiliate, including U of M Health West and U of M Health Sparrow, without first establishing formal agreements. U of M faculty should be aware that although U of M Health West and U of M Health Sparrow have U of M in their names, they remain separate entities in research, requiring all the agreements that would need to be established for research between any two separate institutions. This includes, depending on the study, the subcontracts, data use agreements, transfer agreements, and the like. With regard to EHR access, U of M faculty should be aware that under National EPIC rules, it is not permissible to assess EHR data in Care Everywhere for research if it has not already been accessed for clinical care. Specific agreements must be established to assess EHR data for research at the affiliates, including U of M Health West and U of M Health Sparrow. The U of M Health Statewide Clinical Research Partnership, or SCRIPS, team has been established to support researchers in the various administrative and regulatory requirements that need to be addressed in order to do research with the affiliates. And finally, to request support, submit an intake form on their new website, which can be found under Clinical Research on the Cores and Resources page of the Medical School Office of Research website. This page will be continually updated and expanded with additional information as sites are needed and workforce developed. Associate Dean Julie Lemang is in the audience if we have any more questions about the use of the access of research through these affiliates. Okay, I just want to do 2 shout outs of notable events upcoming soon. The Research Palooza, which is this Wednesday from 11 to 2 in the driveway outside the MSRB buildings. It's an amazing event. I do encourage everyone to go. Should be also weather just like today. And February 27th, 2025 is a Dean's Lecture. It's also the year that we have our 175th anniversary of the medical school, which is amazing. The theme this year is genome research to gene therapy, and the keynote would be Nobel laureate Jennifer Doudna. Jennifer Doudna pioneered work in CRISPR gene editing. Okay, I'm on time. Okay, we're going to move on to the 3 faculty. So it's my pleasure to introduce them and thank them very much for agreeing to be up here. So the first faculty member is Michelle Hastings. She is the Pfizer Upjohn Professor of Pharmacology, Director of mRNA Therapeutics, Center of RNA Biomedicine. Dr. Hastings is studying splice switching antisense oligonucleotides, or ASOs, which have emerged as effective therapeutic molecules that can modify gene expression by modulating pre-mRNA splicing. Dr. Hastings?
Thank you, and thanks for inviting me to talk today. It's really a thrill and an honor to be here to talk to you about the work that I'm doing and really the purpose that I came here to University of Michigan, which was to direct the new RNA Therapeutics Initiative. So I've only been here about a year, but I've really been overwhelmed in a really good way with the amount of interest and excitement around this new technology in RNA therapeutics. And while 5 minutes is a very short period of time to tell you about all the things that we are doing and want to do, I wanted to really just touch on a few things in particular. And I just want to start out with a story. I had an experience the other day. I was in the airport here in Detroit, And I was sitting waiting for the plane, and there's I'm sure you've all seen them, the screens that are there, that there's an ad campaign from Michigan Medicine, Michigan Answers. And I was sitting there and the screen flashed up and said, Michigan Medicine, curing the incurable. And I thought, that's pretty neat. And then the next one, a few slides more, and it said, Michigan Medicine, creating, discovering the future of medicine every day. And this was particularly meaningful to me because I was on a flight to go to North Carolina, where I was going to be present for the treatment of 2 young girls with a life-threatening neurodegenerative disease. And they were getting treated with a medicine that had been created, discovered, and developed in my lab. And this was really seeing that flash up and coming from Michigan and seeing that, yes, that's exactly what we're doing. We are creating and discovering the medicines of tomorrow, the future of medicine. And that was a first for me, but I know it won't be the last. And that's really the reason that I came here, and that I want to work with the scientists and the physicians here to do this, to do this more. This was children that would never have had an opportunity to have a treatment if it weren't for the advancements that we're seeing and that are possible today. And so what am I talking about? So I want to talk about the science, just a couple of minutes about the science, a couple of minutes about medicine as it relates to the science, and then the patients, which are really, at the end of the day, why we're doing this. So the science is RNA. So RNA is really the workhorse. The DNA protects the information of our genome in the nucleus, but the RNA is the message that goes out and really tells the cell how to make the building blocks of our organism. And we now have the ability, and we have an understanding of how to make RNA that can mimic the natural RNA and get it into our body. We all evidenced that with the pandemic, where we were able to deliver a life-saving RNA to protect our bodies against a virus. We also have, in my lab, we're working on small synthetic pieces of RNA that we can deliver very precisely and accurately to exact sequences in RNA, and acts more kind of like a volume knob, a control switch, where we can turn up and turn down the amount of that RNA that's being made in order to overcome genetic diseases. For example, we have people here that are doing amazing technologies around gene editing and changing our DNA using RNA molecules that can then one day cure disease. We have people working on next generation mRNA therapeutics. And it's really exciting, just the new people that are coming. We have a new investigator, Anna Mara, that's coming on delivering genes via viral gene therapies. So we have a massive tool chest of technologies that we can develop. And one thing that has really made this important, and this brings me into the medicine side of things, is that the FDA is recognizing these types of technologies that are coming, and they're beginning to see them as platforms, making the timeline and the cost from going from a discovery to a medicine shorter and more accessible to people even in a basic research lab like what I did, which is exactly what we did for the children that I talked about in the beginning of this. Within 1 year, we were able to do all the studies that we needed to do in order to make this drug in a very fast time, because the very basic platform of these RNA technologies are similar and viewed as relatively safe, with the appropriate, of course, testing that we had to do. And so that has been really a game changer, and why we are eager to work with other labs and physicians to now identify other approaches that we can take. And so that brings me to patients. I'm working with physicians. In particular, a starting place is with neurology, where my co-director, Peter Todd, is a faculty and we've started a gene therapy action board committee, where we are evaluating. Patients get their genome sequenced when they come into the office, and then we have a board that evaluates that and determines is there a possibility for an RNA therapeutic that could could help them, and we're evaluating that, and we are, in some cases, able to start them down that pathway towards developing a therapeutic. And so these are the types of things that we're doing, among many other things, and we know it's the future of medicine, and it's happening right here with the right resources, and we've had so much support from the school already. We're really excited about what's happening.
Thank you, Dr. Hastings. That was amazing. We'll move on to Michelle Kallenberg. It's the Giles Bowles and Dorothy Mulkey Research Professor of Rheumatology and Vice Chair of Basic and Translational Research in the Department of Internal Medicine. Dr. Kallenberg's clinical work is centered on the care of complicated lupus patients, and her independent research laboratory combines translational approaches using patient samples and murine models to uncover the mechanisms that drive lupus and other autoimmune diseases. Dr.
Thanks,
Kelly.
Carl. First, I just want to say, Michelle, your work is amazing. And this RNA Therapeutics Center, is just phenomenal. So really excited about it. And I'm hoping we can bring some autoimmunity stuff to the center at some point here in the near future. So my daughter is a huge fan of The Lord of the Rings and The Hobbit. And Bilbo Baggins wrote a story, as you may know if you've seen the movies, A Hobbit's Journey There and Back Again. And I feel like my research is kind of in that realm where we do what I like to think of as the triple B, which is bedside to bench and back. And a lot of the work that we do in our lab is really informed by the clinical presentations of patients with severe autoimmune diseases. And I was very motivated as a young physician scientist to go down the path of working in lupus in particular, because patients are really struck down in the prime of their life. These are women in their 20s and 30s who are just finishing college, who are trying to start families, who are getting their careers off the ground. And they get blindsided by an autoimmune disease that happens to no fault of their own and can cause very devastating consequences. And when I started 10 years ago, we really had very few treatment options besides blasting them with chemotherapy drugs to try to get their autoimmune disease into remission. And so, in thinking about the clinical problems that these patients face, while there's been two areas that we've really been focused on in our work. One is that we've had no good way to understand how a patient is going to respond to a various treatment. And what ends up happening is that we end up prescribing drugs as dictated by insurance companies because they will tell us what we can prescribe because we don't have data to tell the insurance company what this patient needs. So that's been a very important piece that we've been trying to tackle. And the other piece is that there's a very interesting phenomenon in lupus where patients get sick by just going out into the sun. In many of our autoimmune diseases, patients get treated with sunlight. So psoriasis, for example, we put patients under UV light in order to calm the inflammation in their skin down. But in lupus, patients actually get rashes from being exposed to the sun. And if they get enough exposure, they can actually have a big systemic flare of their disease and have their kidneys get inflamed and all sorts of things happen to them. And so this has been a very interesting question for us. So those have been kind of like the two driving principles in our work. We were very fortunate in 2019 to get one of the first Taubman Institute Innovative Program grants, which allowed us to set up a very robust clinical research infrastructure to recruit patients and start to develop longitudinal cohorts. And I think this is a really important piece to the research infrastructure here at U of M, is that you have to be able to collect patient samples and patient data in a way that will let you study them to understand how they respond to drug. Because we can't just say this is what you need. We have to understand why patients need certain medications. And so that's been something that we've been building out, and we're getting to the point now where we can start to divide patients into responders and non-responders to various drugs and understand what the drivers are to help us develop a personalized medicine approach to these patients. The other piece, the photosensitivity piece, the sensitivity to UV light, we've done some, I think, really exciting understanding the impact of various inflammatory molecules on how sunlight reacts with the skin. And we actually just finished a clinical trial sponsored by the NIH looking at a drug that's approved for rheumatoid arthritis and how this may impact photosensitivity. And so again, we've taken it back to the bedside from starting from a clinical observation all the way through. And hopefully this will lead to a larger trial where we can test this and hopefully let patients go back out to their kids' soccer games without worrying about setting off a disease flare. So overall, I would say, there's a lot of progress that's been made, and these personalized medicine approaches are really going to transform how we can care for patients, save time, save money, and hopefully get patients back to their lives with much less hassle and side effects down the road.
Again, very amazing. Thank you, Dr. Kallenberg. Okay, the third speaker is Dr. Steve Parker, Professor of Computational Medicine and Bioinformatics and Director of the Epigenomic Metabolic Medicine Center, or EM2C. Dr. Parker integrates genetic and genomic data to dissect regulatory mechanisms for diabetes and related metabolic traits. Steve was recently given the Outstanding Scientific Achievement Award from the American Diabetes Association. Dr. Parker?
Well, thank you so much for the invitation to speak here. It's really humbling to be up here with such superstar faculty colleagues. I think if my wife and I have another daughter, we're going to name her Michelle. Just kidding. We're done having kids. We have a 1-year-old in the house. So today, I'm super excited to share with you how integrative data analytics and the study of the epigenome are transforming our understanding of disease. It's going to sound a little bit audacious, but bear with me for 4 more minutes. So imagine a world where we can predict and prevent common diseases before they even appear, thanks to the powerful combination of cutting-edge technology and innovative research. Now, a little bit of historical context. 20 years ago, in 2004, almost to the month, the human genome sequence was declared finished. I was working on that at the time. I was at the MIT Center for Genome Research. Now it's called the Broad Institute, and I was a genome closure data analyst. It was a super exciting time to be doing science. There was this race between the public sector, led by Francis Collins, and the private sector, led by J. Craig Venner, and both published at the same time, even though the public sector did a much better job. No bias. I was on that team. And I'm happy to walk you through the science of that. It's actually true. Shortly after that reference genome was published, the community moved on and we started exploring population variation and how genetic variation is associated with disease predisposition. Shortly after that, the first GWAS was published, or genome-wide association study, and that is a type of study that associates genome variation across the population with diseases and traits. Type 2 diabetes, which is one of the diseases I study, was among the first published, and there was just a handful of loci. Fast forward today, T2D, one of the largest actually disease GWASs ever published with over 1 million individuals, has over 1,000 loci that are independently associated with disease predisposition. And I want to be inclusive here. Diabetes is not the only trait for which GWAS has been done. There's lots of diverse diseases and traits. Collectively, there are tens of thousands of regions of your genome that encode disease predisposition. The tricky thing is that the vast majority, 90% of these regions, are non-coding. That means it's really hard to identify what's the causal gene, the effector transcript, and even more important than that, or as important, what's the causal or effector cell type? What cell type is that genetic variation acting through? So, since then, the community has made tremendous strides with technologies like single-cell sequencing, which allows us to examine diverse omics profiles, the genome, the transcriptome, and in between those two, the epigenome, which I think is the most critical layer to understanding causality. And it allows us to see the world like looking through a high-resolution microscope. So today, we are starting to generate, like we did from human genome to variation across the population scale single-cell data sets that were unimaginable just a few years ago, much like we moved again from the reference genome to understand variation. At the University of Michigan, we're investing heavily in these advanced technologies with comprehensive core facilities that set the stage for truly groundbreaking research. Collaboration is key for success. I currently have about 15 different grants in my portfolio that span foundations and industry and NIH, including R mechanisms, U mechanisms, P mechanisms, all kind of stuff, and collaboration is essential for all of these. So what is the epigenome? It's the layer between the genome and the transcriptome. Gene regulation happens through this layer, and it can be very context specific. By context, I mean different genetic variations across individuals, but also different environmental variations. So it's critical to build up these population scale data sets to understand how genetic variants percolate through the epigenome and are caused on the transcriptome, so folks like Michelle can design targeted therapeutics. So we're using AI to integrate epigenome, genome, and transcriptome data sets to better understand disease mechanisms. And looking ahead, I think the integration of AI in biomedical research offers even more promise. Imagine using AI to analyze vast data sets, revealing patterns that can lead to new treatments and preventive measures for diseases like diabetes. So I want to say thank you, everyone, for your time and attention, and I look forward to seeing all of your exciting research.
I'll just say those are three fantastic presentations, and how you could squeeze everything that you did into 5 minutes is even more amazing. But thank you so much, and I'll turn it back over to Carl.
Okay. Keith, you want to take this?
Yeah, well, let's start and see if we can have a bit of a conversation here. We have a couple folks in the back with microphones, so would love to have folks raise their hand if there's a question you want to ask of anyone on the panel on the stage. And I would encourage you all to do that. It's not that often that we have such amazing speakers all in one place. All right, there we go. Thank you.
Hi, I'm Joe, and I just have a question for our amazing researchers. What you're doing generates vast data sets. How are you managing them, and what is Michigan doing well, and where can Michigan do better?
Yeah, maybe I'll start there. My primary affiliation is in Department of Computational Medicine and Bioinformatics, and I have secondaries in human genetics and biostatistics. So we operate on the hundreds of terabytes, a few petabyte scale of data generation and analysis. I'll say I arrived at Michigan 10 years ago in 2014. The Great Lakes High Performance Compute cluster was not in existence upon my arrival. So we took some of our own money and invested in our own resources. Then Great Lakes came along, and it's been amazing. And now Great Lakes is evolving. For example, a lot of the AI and kind of deep learning methods that I alluded to in that little 5-minute spiel, they require these really high performance GPUs, not CPUs. And Great Lakes is continuously adding more GPUs to the queue. So we're not waiting in the queue. We want answers like yesterday, always. And so waiting in the queue is not cool. We want more GPUs, and Michigan is really great at doing that. I guess if I had to be really picky and say what's an area that Michigan could do better, is that data storage is now becoming a problem. So the compute is fantastic. It's highly subsidized by the med school, and it makes us very competitive in grant proposals. The storage is exceedingly expensive, especially if you're talking about data sets that are on the scale of hundreds of terabytes to petabytes, right? So I think that's an area of growth and development, and I know folks are working on that, and I guess that I'll give the other folks a chance to chime in now. But it's a great question, thanks.
Yeah, I would say, again, the Great Lakes is amazing, and being able to access those is great. I would say one other opportunity for improvement that I would argue for is, now we have like the most amazing microscopy facilities here, and we're getting the most beautiful images, which are huge in size. And none of our standard issue computers, desktops that are core image, can actually even open a single picture. So it's a real challenge to get people to be able to do their data analysis on our standard computers, because they don't have enough internal memory to even like open a file. So you have to plug into these outside networks. And now with the restrictions on VPN and those types of things, there's just like multiple layers of difficulty getting to data analysis. And so I think that's something we need to think about. How do we improve that and allow our people to be able to do work all hours of the day sometimes.
I don't have a lot to add. I'm still transitioning from previously, so I haven't had the same experiences, but.
Thank you all. Other questions? I see, yes, another hand in the room, couple hands. The woman here in the back right, and start.
Hi, this is Vicki Blanc. Thank you for such tremendous work. I really appreciate it. I loved hearing all about it. So thank you for doing what you're doing. Each of you mentioned collaboration as important to your research. I'm wondering if you could expand a little bit more on how you set up collaborations at Michigan. What's beneficial to you? And in the spirit of the previous question, what can we do better?
I can start there. That's one of the best things about moving here so far is how many people have just really reached out through email and contacted me to start collaborations. The amount of internal opportunities for getting small grants for pilot studies has been really great as well. The physicians also have been really eager to work with us, and so we've created a lot of working groups around physicians and basic scientists. And so I think that cohesiveness across the bench lab space all the way out to the clinic is really amazing. And it's one of the reasons that I came, and I have not been let down. It's been great. I mean, like I said, the number one thing is getting the funding, the pilot studies funded when we're starting from nothing, but we've had a lot of opportunities in that direction as well.
Thank you. There's a question over here on the, yeah.
Yeah, beautiful presentations. I'm Niels Woter from chemistry, and I have a more strategic question, I guess. So I noticed that both Steve as well as Michelle Kallenberg, of course, referred gratefully to Michelle and RNA therapeutics generally. And putting those dots together, I feel there is clearly a connection here where Steve's GWAS studies and similar ones would, as he said, generate data that then would allow RNA medicines to be developed based on sequences, and then those being applied to all sorts of clinical complexities and challenges like Michelle Kallenberg alluded to. So I'm wondering how much of the strategic connecting of dots is actually going on behind the scenes.
I would like to just really emphasize the importance of your question. And I know that there is a group working on cellular and gene therapies, and it's been sort of an organic coming together of RNA and people doing CAR T studies. And what they're really looking for is investment by the institution. So I'm just going to put a plug out there that I think there's a really strong group of faculty who are organizing and getting all of the right pieces together. And what they really need is a big capital push to make it happen. And there's a few infrastructure pieces that need to come along with that, including maybe expanding some of our synthesis capacities and other things. But there's a very strong group of faculty here pushing for that. So I'm just going to put that plug out there.
Yeah, and we've been working with the cell and gene therapy group as well as, obviously RNA therapeutics is one arm of cell and gene therapies. And so we've been working alongside them to try to get some of the things we need. For me, a lot of people come and exactly want to have RNA therapeutic molecules designed and synthesized. Right now for us to go out and buy them, even for research grade, can be somewhat cost prohibitive, even though there is the potential to make them for 10 times less and much faster. And so that's one thing we want to bring here to really fill that gap in terms of enabling basic science to work in this area. So.
I guess I'll just add, Niels, that's a fantastic question. I think it's related to the question that Vicki posed about collaboration, because the landscape here at Michigan is so collaborative. There are so many experts here. You can basically do a project in anything you can dream of. But that's also hard, right? Because it's a very complex landscape. So strategically connecting all of those complexities across experts is a very difficult problem. I know Carl invests a lot of mental energy in thinking about this, and Steve does too, and there's really good institutional infrastructure to leverage those connections. Like if you think back to like the M-Cubed initiative, where you needed 3 different investigators to come together to fund a project. So I think there are lots of kind of strategic leadership ways to induce exactly what you're asking about. And we're not going to solve that right here on the stage, but I just want to acknowledge and put a pin in the Michigan landscape is beautiful and collaborative, but that also creates complex structure across research enterprises and people.
And I guess at the risk of maybe piling on that complexity, I'm curious, are there opportunities that we're missing in collaboration across with the rest of campus?
I mean, me personally, most of my collaborations are within the med school. I do have an appointment in biostatistics at School of Public Health, so I do also collaborate extensively with public health folks too. Not as much with engineering or LS&A, and that's not for any kind of purposeful reason. That's just kind of how the research evolved. So I don't know, not to harp on M-Cubed, but I think it was a great mechanism, and it really forced people to go across schools and institutions to collaborate together. So it may be something like that. I don't know.
Just to add to that, so Caleb Smith, who works in the grants office, has done a connectome, trying to figure out how we are connected across campus based on authorship and as well as grants. And college engineering is our #1, and then I think School of Public Health and LS&A follow thereafter. But Michigan Medicine is like the heart of Michigan, no offense to the rest of campus. But we are literally connected virtually all throughout campus. But I also want to add to that, the sympathy, because as we hear this often, that the strength and simultaneous weakness of U of M is its size, right? And we have these wonderful collaborations that can be established, but having people find each other when they need to is often very difficult. So we're always looking for new ideas. It's this idea of not promoting Match.com, but it's the same kind of concept where you're having people get online trying to figure out who is looking for someone else, and can we create some new technologies that help connect people who are willing to partner with research. But any other ideas, let us know, please.
Thanks, Carl. Other questions? Looking for hands? All right, we got one more there, Neil, and then Brian.
Yeah, maybe as a follow-up question to this overall strategic thinking, I think it requires, and I really appreciate that, strategic vision from leadership. And of course, we now have that also from the highest levels in the Vision 2034. We heard about the Innovation Hub and the Conference Center here on NCRC in the future. So that provides, I think, opportunities to connect people more. And I was hoping to hear a little bit more about, for example, what will go, what's the vision behind the innovation core or center, and how will this all be connected at this higher level where, Vision 2034 might make strides.
Yeah, Marshall.
I can comment at a very undereducated level about Vision 2034. I've been involved in that, and I think that that's a very broad vision. I think the innovation approaches there are a key component of it. I know President Ono is absolutely dedicated to that. There are going to be very substantial resources committed to Vision 2034. I don't remember exactly the numbers, but I think it's $100 million a year for 10 years, something of that order. And as you know, it's just been announced there'll be a structure to trying to identify those areas of research that are most critically needed. And so research across the university is a huge thing because it runs all the way from very basic discovery research to social sciences and other areas, and in many different areas. But I think that just because of the cost and the potency of the research community, not just in Michigan Medicine, but across the university, I think that will get a large portion of the investments. And so I guess the one thing I can say without really, it seems like I haven't really said anything, but the one thing I can say is if you get called upon to participate in this visioning, which will be starting really this fall, later this fall, please do. Every committee is a new sink of time, I know, but please do participate in that, because I think your input is going to make, that's the whole idea of Vision 2034, is a grounds up input, but it's going to really help make decisions as to where investments will occur over the next 10 years. So it's a great question. I'm sorry I couldn't give much of an answer, but that's sort of where things stand. Oh, I forgot, this is totally unrelated, but I forgot in my comments to say that this discussion day isn't being live casted, but it's recorded, and it's going to be on One Michigan Medicine and on our headlines. So it'll be available for anybody that wants to look at it, including your friends and et cetera.
Hi, Brian Athey here. Thanks so much for this great vision. I wonder if you can think about translation. Maybe Michelle Kallenberg and Julie can talk about that. We have all the basic science. We want to get it to the patients. What would be the plan for the translation of this, and how can we help?
Well, I think there's a few challenges. First of all, we have amazing clinicians here, and in terms of taking things back all the way into the clinic, I think one of the things that's perpetually in improvement mode right now is our clinical trials enterprise, and sort of how we fund a robust and thriving clinical trials program. And some places within the medical school do it really well, and some are still having challenges implementing. And so I think we need to continue to think about that, and how do we make that sustainable. So that I think is really important, and providing time for clinical faculty to participate in that piece, I think, is important as well. So I think that's one thing. And then the other piece of that is sort of like before we get to that point, and maybe this goes to the vision, and something that I think we need to maybe think about strategically as well, is we're really good at generating data. It's the clog in the pipeline is the analytics of the data, and how do you extract meaningful information from that data. And that's really where you need collaborations from the clinicians and the scientists and the analysts to all come together and understand, what does this actually tell us? Is this feasible, and does this make sense? And could we actually do this for a patient? And so, how do we invest in that part of the collaboration, I think, is also something to think about. And how do we get the analytics to catch up with our ability to generate the data? Because I have data that we generated 4 years ago, and we're still slogging through the analysis, right? I wish it was faster, but that's just how the reality has been. And so how do we speed that up, I think, is a good challenge for us to think about too.
Yeah, and I think that having just gone through this process for the first time, really translating something from the bench to the bedside in an ultra rare, very personalized manner, I've learned a lot about the process, really coming from sort of the opposite direction that it normally comes, which normally you have a big pharma that might license something and develop it, and you're kind of waiting with your own basic research for that to happen, and if it doesn't, then you sit there. But we don't have to do that anymore. What I've seen is that we have the ability and the power to be able to generate that interest. In my case, it was a foundation, the family foundation, that raised the money for us to be able to do that, to drive our research forward. We got experts in all of the different areas that we needed to bring that forward. And so I think we have to start thinking about drug development in a different way also, not just the traditional path forward. And here at Michigan, like I said, we've developed a genomic medicines board, much like a tumor board for genetic disease that comes forward. And our real intention is to find answers for these people that otherwise would never have a solution. I think we've all, if we haven't yet, we will one day be in the doctor's office and get that hopeless diagnosis that there's nothing that we can do. But now that really is changing. I think with genetic medicine, as we've heard about, we're starting to learn and understand how we can actually get in there and change things in very precise manners. And that opens up huge potential, and we have to find new ways to drive that forward. It can't rely on limited amount of pharma who want maybe big, large, broad diseases. So that's sort of my take on translation.
Just one other thing, and Brian, that's a great question, because I think this also speaks to what we talked about earlier about collaboration. The vast majority of our departments are clinical, and those scientists that sit in those departments have clinical partners that they can reach out to. And they can take their basic science discoveries and then maybe more easily translate them, but that's not true for all of our departments. And so speaking to that, I know Ms. Shar, I'm not gonna put Julie on the spot, but Ms. Shar offers a lot of services to help people understand how to go from basic science discovery through translation, through clinical, as well as implementation sciences. And if you go beyond that into policy, that's the nice thing about Michigan. I'll give us that we are a full stream operation. Okay, I think everyone understands that, and I hope you really appreciate that. But back to this point, if there are some holes that we are not aware of, please let us know. We would like to kind of make sure that everyone has the ability to go from basic through translational through clinical.
Other questions? I have one for those of you who are working in large data sets, and it sounds like increasingly there may be opportunities to use Gen AI to help with the analytic piece that, Michelle, you were just talking about. But every time I hear that, I also start to wonder about systemic bias in the data and sort of black box interpretations of the data that may introduce more bias. And I'm just curious, as you all think about your work, how do your perspectives on health equity and how do you go about guarding against systemic bias in the work you do?
Yeah, so maybe I'll start there. I think this builds nicely on the previous discussion around data analytics and kind of like large-scale data sets. So we just started a new grant about a year ago. It's a U24 mechanism where the whole idea is to create this like large-scale Gen AI knowledge base about the human pancreas to aggregate all data in there. And we're very concerned about all sorts of biases, like you'd mentioned. One area of bias is kind of like data quality bias. So the initial steps are only put in the highest quality data that's based on rigorous standards, right? But also other systemic biases that our team, it's a multi-institutional team doing this, but our team's not broad enough to probably recognize everything. So the idea is to have a community board and get feedback constantly from the community board in an iterative process. And I think this is such a timely discussion of this particular topic, because just last week I was, had a meeting for this consortia, and we are in constant discussions with lots of NIH program officers. And recently, the leadership funding this is through is NIDDK, the Diabetes Institute. Recently discussing in front of Congress and getting congressional feedback, and the feedback from that I got is basically everyone in Congress is incredibly enthusiastic about AI and biomedical research. That is now starting to trickle down to enthusiasm from NIH. So I think on the very near horizon, there's going to be lots and lots of funding opportunities for AI and biomedical research. And I already see it happening in NIDDK, and if the other institute leadership are anything like the DK leadership, they probably are having similar meetings with congressional leaders who are going to push this. So I think Michigan is very well positioned to capitalize on this, and a little bit of forward thinking might position us even better.
Yeah, I agree with all that. And I think a couple other challenges, at least with the health equity piece, is that we still have a lot of patient populations that are underrepresented in our data sets. And so one of the things we've been doing, at least in the lupus world, is working with community advisory boards and trying to understand sort of what are the barriers to participating in research from patients that are underrepresented. And also in our PO1 that hopefully is going to be funded, they say it will be, but we built a, just if Congress doesn't implode and we still have an NIH after the election. So one of the things we actually built into that center is actually funding for the Community Advisory Board and building patient researcher engagement sessions so that we have patients come and learn about the science, and the researchers can also meet patients, because not everybody has the luxury of going to clinic and talking to those patients. So we want our basic researchers to understand what it's like to live with lupus. And so they can learn from the patients too, like what's important to them, how do we design studies that they want to participate in. So I think encouraging people to think about these health equity pieces as we write large center grants and find other sources of funding, I think is important. The other piece with data that I think is a national problem, a world problem, not something we could probably solve here, is just the annotation of data and making sure that we have good clinical characteristics associated with the data we generate. Because if you don't know it's a untreated patient versus a patient who's on steroids, and you say both of these patients have disease X, and then you look at the data and you're like, well, these patients don't look at all like each other. If we don't understand what is the clinical things about that patient when that data is collected, it becomes a little bit useless or confusing and can like muddy the water. So I think we really have to think nationally about all of this data that we're generating and then making publicly available. How do we annotate it in a way that it's useful to the research community as a whole? That's a big problem, but something that we always have to keep in mind.
Thank you.
Just to add to that, yes, NIH is very aware of the limitations in the clinical trials that exist out there, which is our existing data. And so we're way behind in our data collection. A lot of the clinical trials are very biased towards certain populations. Okay, we have to be very cognizant of that when we build these out. But U of M, as a world leader in research, this is our opportunity to lead the field. The task force that Brian Athey is leading right now will help set those guidelines so we can move forward with acceleration and confidence that we're doing this correctly.
Thanks. Marshall?
I have a comment and a question. So again, this has been like the most fun I've had in a long time at work. So thank you. And I've gotten some ideas as I'm sitting here listening to you in one area where I think we may be able to find funding for some of the things that you're interested in is through philanthropy. And we're just getting ready to start a new campaign, and so I'm deeply involved in that. And so the ideas that you've given me today are really helpful. What I want to ask is a brief explanation for something that Steve Kunkel's been trying to explain to me for a couple of years now, and that is a better understanding of epigenomics. And so to take, I'll give you an example. So to take a broad disease, a polygenic disease like diabetes, type 2 diabetes, how does one go about identifying those epigenomic markers, or whatever you call it, marks, that are most important? And then how much of an ordeal is it to then, perhaps in a cellular fashion, to test those with an RNA approach or other approaches to see if you're on the right track? And then final part of that is, have these epigenomic factors, because I know they get somewhat translated through generations, have they been around forever, or are they something that comes to a family and then they pass it to their children and their grandchildren, and that's the end of it? Is that too much for a couple of minutes?
No, certainly no.
No pressure.
Yeah, that's a wonderful question. It's very complex, and so let me try to give some tidbits that I think will answer it. So the current latest T2D GWAS came out a few months ago, over 1,000 different regions of your genome predisposed to disease. And really, it's difficult to know how they act, right? Diabetes is a complex disease. It works through skeletal muscle, adipose, pancreatic islets, the brain, all types of different tissues. And just knowing those genetic associations don't tell you which tissue or which target gene we need to get to. So this is where these single cell epigenome profiling technologies come in, right? Most of these genetic associations are non-coding. The genome is folded up in complicated ways and has all these little bumps in there, and we need to figure out which of those are open and doing gene regulation in specific cellular and tissue context, right? So that's where single-cell epigenome profiling comes in. Then you can kind of compare cell-specific maps to these genetic risks and say, oh, this locus number 578 looks like it's operating through skeletal muscle, whereas locus number 579 is operating through pancreatic islets. Then you can go partition that across the population and say, oh, an individual over here, you have diabetes predisposition, not just complex diabetes, but it's really, you have skeletal muscle dysfunction. And someone over here has adipose biology dysfunction, and someone back there has pancreatic islet beta cell dysfunction, right? So you can start partitioning people into not just these tissue agnostic, you have diabetes predisposition, but now you can have pathway specific, tissue specific, physiological specific buckets of individuals and kind of triage them appropriately. So the question is, have these been around for a long time? Right now, GWAS is only well-powered to detect common variations. So these are variants that have been around for a little while, but that's not to say that there are other more rare variants out there that are important too. And just to hopefully tie this all together, common predisposition is largely in the regulatory landscape that we see in the epigenome. But if you look at rare forms of diabetes, that is coding variations, high impact genetic variations. And we're starting to see convergence of, okay, there's a coding variation for this transcription factor that's very rare form of the disease. And then the binding sites where this TF lands, that's where you have these smaller effect GWAS type variants that perturb the binding site. So you can see the network coalescing, and now we're starting to understand what are the kind of peripheral genes and the hub genes, and how do we decide tissue-specific targets for those. So hopefully that was...
I think, and you can think about it in a different way. The way my brain kind of thinks about epigenomics too is that there's probably environmental exposures that people get exposed to in their life, whether it's chronic sunlight exposure or toxin exposure or PFAS exposure. These things change how your DNA works from the time you're born with your genes. It changes things and changes how your immune system's talking and your nervous system is talking to each other. And one of the things we're very interested in, because we have some idea that in lupus, for example, we can take a skin biopsy and grow people's skin cells in culture, and we see that they behave very differently in culture with no other stimulus from cells from a healthy control. So something has happened to the skin over that patient's lifetime where it's not behaving the same. And one of the things we're very curious about is whether some of our drugs might actually help reset them back to normal, because we know that inflammation is a very important trigger for epigenetics. So if you have a cell and it gets exposed to inflammation, it's not going to be the same after, because the way that the genes are accessible has changed, and that's all through epigenetic mechanisms. So we're very interested about how treatments might revert people back to normal, so that it would tell us, how long do we have to treat somebody for? Can we put them back to a healthy state with 6 months or 1 year or 2 years of therapy? And then that way we can make an educated guess about when it's time to take somebody off of drugs. So I think about epigenetics in a slightly different way, but I think also very important for how we treat patients.
And I'm always thinking about targets. So I think, of course, the biggest barrier, I think, the bottleneck to RNA therapeutics is having good targets and understanding what you need to do, because now I think we have so many tools to increase or decrease gene expression in various ways, but knowing whether how much you want to increase, how much you want to decrease, when and where is, of course, critical. So,
great. With that, I think we're coming up hard on our time limit. I want to thank Steve and Michelle and Carl and Marshall for all of your great comments and elucidations of the work you're doing. I will say, as I listen to you all, I think it was maybe 40 years ago, Richard Dawkins tagged genes as selfish, but I see nothing but great evidence of the generosity of the genes, maybe your own genes as well as the genes you study in terms of giving back. So thank you very, very much. And thank you all.